Today, what I'll be doing is I'll be really covering in detail the company, Defense Therapeutics, as well as the projects we're actually running since 2018 in terms of research and development. So Defense is currently trading on the Canadian Stock Exchange and the Frankfurt Stock Exchange. It's also available on the OTC, and there is really a program to or plan to actually go on NASDAQ by Q4 of this year. So what is Defense? Defense is basically a Canadian biotech company developing the next generation biopharmaceuticals for various indications in the fields of immuno-oncology and infectious diseases. So it uses its proprietary platform called the Accum, which stands for the accumulator, simply because this is a platform that allows the accumulation of very important biomolecules, which could be proteins, RNA, or DNA. And really, we're well positioned to achieve data-driven value inflection points with all the progress we're doing, and also with the fact that we are planning to start 2 to 3 phase I trials in Q1, Q2 of 2023. So everything is being run at the moment in terms of GLP studies, interactions with regulatory agencies such as MHRA in the UK, Health Canada, and the FDA to really start our phase I trials on 2-3 different products, which I'll be talking about in a moment. And of course, Defense is very happy to be able to regroup a team of experienced personnel, whether we're talking preclinical or clinical developments, business, CMC, and regulatory. So for instance, we're led by the CEO, Mr. Sebastian Plouffe. We have our CFO director, Joseph Meager. We have also our CTSO, Dr. Simon Baudouin, who's basically the co-inventor of the Accum technology. And we were lucky to have on our board 4 experts, Dr. Plefederer, who's an expert pharmaceutical advisor in Europe, mostly Dr. Riam Shama, who's also an expert in seed investor, Dr. Lobenberg, who's really involved in the regulatory part of our development. And of course, we have also Dr. Mitrician, who's the head of the breast cancer oncology unit, who has extensive expertise in clinical developments and treatments in patients. So as you probably know, there are major challenges when it comes to immuno-oncology and infectious diseases. We all know that there are standard of care that are currently being applied, but unfortunately, the standard of care, which is composed of chemotherapy, surgery, radiotherapy, doesn't necessarily or is not efficient at treating disseminated cancers or cancers that have been metastasizing throughout the body. And so there are multiple therapies that are being developed. For example, we call these targeted biopharmaceuticals, which could be, for example, antibody-drug conjugates. But the problem is these are facing molecular obstacles in the sense that these are entrapped in certain components within the cell, which limits tremendously their potential to really reach the target and induce the therapeutic effects they are designed to achieve. And another point, there are no real anti-cancer vaccines on the market. I know we've heard a lot about vaccines lately, mostly for infectious diseases, but when it comes to cancer, there is a tremendous lack in that field. I know people have spoken about the Gardasil or Gardasil 9 as being an anti-cancer vaccine, but it's really an anti-HPV vaccine. So it's not really a real anti-cancer vaccine. And of course, immune checkpoint blockers are extensively used in the field as a monotherapy, but there is, again, lack of responsiveness that has been reported lately. This is why people are currently searching for new strategies where they can combine the immune checkpoint blockers with these therapies in order to achieve a synergistic effect. In other words, A plus B equals D type thing. So overall, I can say that there's an urgent need for enabling a first-in-class versatile technology that could bypass most, if not all, of the above mentioned limitations. So what is the Accum technology? If it all comes back to this small molecule that is composed of a bile acid linked to a nuclear localization signal. So this whole molecule is really, really small. We're talking about 2 to 2.5 kilodalton in size. Of course, Defense owns the technology platform. And what this molecule does is that it triggers escape of the biomolecule by breaking down endosomal membrane, which is where usually the molecules or biomedicines are entrapped. And of course, this molecule is highly versatile because you can apply it on different antibody-drug conjugates, vaccines. You can even imagine using it in the field of CRISPR, or to deliver any kind of DNA or RNA molecule you wish to have in that sense. So this is an experiment just to demonstrate to you here how does the Accum works. So protein called the enhanced green fluorescent protein fused to the human galectin-3. So this is a protein that can bind to sugar residues that are usually found on the cell surface. But because it's fused to a GFP, if you overexpress it in a target cell, the entire cell will look green because the protein is distributed uniformly throughout the cell. Now, what is interesting is that if you add to that cell the Accum molecule, then what ends up happening is that the Accum is going to break down the endosomes, and that will allow these proteins, which were uniformly distributed, to relocalize within the endosomal lumen. So within the inside of the endosome, where you can have access to glycoproteins or proteins that have sugar residues on their surface. And that's why you can move from a cell that is entirely green to a cell where you can see distinctive punctines all over the place. So these are damaged endosomes that are becoming highly plastic. So you can get in and out of these endosomal structures. So if you would like to learn more in terms of how this technology could be applied and how we used it in the past, I strongly encourage you to read a review that we just published in the journal Molecules, which really talks about the developments or the use of the Accum, whether it's in the antibody-drug conjugate world or even in the protein field, and what we've done so far with this molecule. So in terms of the pipeline, Defense has currently 5 products in development. So we're actually developing, in terms of cancer, a DC and protein vaccine. So this is a dendritic cell-based vaccine, which I'll be talking about shortly, targeting melanoma. We also have a vaccine targeting cervical cancer. So this vaccine is actually very interesting because it targets both it could be used as a prophylactic vaccine, so protecting from the cervical cancer, or as a therapeutic vaccine in the sense that it can treat cancer. We also used the Accum platform to develop a molecule called AccuTOX, which could be used directly as an anti-cancer molecule, and this is currently being used or for the indication of both melanoma and breast cancer. We have an antibody-drug conjugate that is currently being investigated in non-GLP studies. We're going to be starting the GLP studies very soon against breast cancer. And we also have a lot of studies conducted on another protein-based vaccine targeting COVID. So what I'll do in the next, I want to say 30 minutes or so, I'll be going over an example of each one of these DC vaccine, protein vaccine against cancer, the AccuTOX and the ADCs. So let's start with the AccuVAC-D001, which is a DC vaccine specifically designed for solid tumors and in this case against melanoma. So as you probably know, there is currently one dendritic cell vaccine that is FDA approved for the treatment of prostate cancer. It's being actually generated by a company called Dendreon. The vaccine works as follows. So you collect the monocytes from that patient you want to treat. And once you have the monocytes ex vivo, you can then treat them with a fusion protein containing the PAP antigen fused to a cytokine called GM-CSF, or granulocyte macrophage colony stimulating factor. And what that molecule does is that it delivers your antigen of interest as well as a signal to promote the differentiation or maturation of these monocytes into activated antigen-presenting cells or activated dendritic cells, which would then, when infused back in the patient, trigger an activation of the CD4 and the CD8 T cells to be able to mount an immune response against the tumor. Unfortunately, when that vaccine was put on use in prostate cancer patients, Spilucil T, which is the scientific name of the vaccine, did not lead to a better outcome, to have a good outcome. In other words, we were able to observe 4 to 6 months improvement in survival, but that's not really what we were aiming for, in the sense that you would expect that vaccine to promote at least 1 to 2 years of survival, which was not the case. So with this in mind, we said to ourselves, well, can we use, in this case, the Accum technology applied to the DC vaccination to break down the endosome and improve the T cell activation of both CD4 and CD8? So the answer is yes, we're able to achieve that. We published it in a very prestigious journal, the Cell Report Medicine, where we described a dendritic cell that was treated with an antigen linked to Accum, and we've demonstrated that we can enhance antigen processing and presentation. We've demonstrated that these pulsed dendritic cells, which are basically the cellular vaccine, elicit potent effector T cell responses. We've demonstrated that we can apply it to an allogeneic DC preparation. And in other words, these are DCs coming from genetically unrelated individuals. So it's an off-the-shelf allogeneic product. And we've demonstrated elegantly that we can increase the CD8 to Treg ratio. So CD8 are the cytotoxic T cells that are known to kill the tumor, and the Tregs are the immune suppressive cells that are usually recruited by the tumor to shut down the immune response. So if you have an increase in CD8 and a decrease in Treg, you're basically in business. So this is basically one of the experimental results we have. So what we did is we transplanted a solid tumor subcutaneously in animals, and once the tumors appeared, so it became palpable, we then delivered 2 doses of the DC vaccine. So 1 dose 3-4 days after the implantation and 1 week later. And in parallel, we delivered the immune checkpoint blocker anti-PD-1 at 200 microgram per dose. So this is basically a therapeutic setting. In other words, you transplant the tumor first and then you treat. So it really replicates what we can see in the clinic. So as you can see here, the red group here is the group that received the dendritic cells treated with the EL4 with lymphoma lysate that contained the Accum molecule in combination with the anti-PD-1. So in that group specifically, we were able to achieve 30% complete responses and 40% partial response. So a partial response meaning that the tumors were kind of controlled, so they were not cured, they were not growing, but they were there. A complete response is when we have cure, so the tumors are actually regressing and disappear down the line. And if you follow up these animals up to day 54, you end up realizing that 70% survival was achieved with our vaccine. Now, this is tremendously good, because if you compare it, for example, to what's out there being developed so far by other publicly traded companies, you can see that their allogeneic DC vaccine really just delay death of these animals by about a week or 2. Whereas in our case, we're able to observe really cure and partial responses occurring in animals. So the other point I want to mention here is that if you look at the vaccine on its own, shown in blue, there is a slight delay in tumor growth. And if you just look, for example, at the immune checkpoints, the anti-PD-1 delivered alone, there's no real effect. So the take-home message here is that when you combine both the anti-PD-1 immune checkpoint blocker with the vaccine, that's where you end up having synergistic effects, where you can really amplify the immune reaction and lead to a therapeutic effect that you're looking or expecting down the line. Now, mechanistically, we looked at the tumor vicinity. So we took these tumors out of these animals and we started, looking at different immune cells. So we looked at myeloid cells, CD4s, another type of myeloid cell, but in this case, the dendritic cells, endogenous dendritic cells, CD8, NKs, and CD19. CD19 are the B cells, NK cells are natural killer cells. And what we noticed is that the animals that have received the vaccine with the immune checkpoint had a tremendous increase in the antigen presenting cells in the tumor, which is very encouraging because it shows that the allogeneic vaccine is also relying on endogenous antigen presenting cells to help them mount the immune response. That combined to an increase in CD8 is remarkable because these are the cells that will eventually destroy the tumor in an antigen specific manner. And of course, you have a significant increase in the NK cells, which are also involved in eradicating tumors in this context. But as I mentioned, what's really impressive is that if you look at the ratio of CD8 to Tregs, you can notice that the group that received the Accum lysate DCs in combination with the anti-PD-1 has a significant improvement in that ratio, which is exactly what we're looking for when we're designing a therapy based on the immune system or an immunotherapy. So you want to make sure that the CD8 are overactivated as opposed to the regulatory T cells. So mechanistically, if I can summarize the whole concept behind our antigen or our allogeneic vaccine. So once you deliver the vaccine along with the anti-PD-1, you transform the tumor microenvironment, you trigger production of IL-6, interferon gamma, MIP2, and MIP1B, which are pro-inflammatory chemokines. These will attract CD8 T cells, NK cells, of course, as well as endogenous antigen-presenting cells, and immune cell recruitment is then taking place and leading down the line to amplification of the immune response against the tumor, which will down the line trigger or elicit their regression or rejection. So far, we are preparing our phase I trial in the UK. So this is a 3+3 design where we are planning a dose escalation study in combination eventually with the immune checkpoint blocker once we identify the best dose. This is done in collaboration between the Birmingham University for the DC manufacturing and the Christie Hospital in Manchester for the location where the phase I trial is going to be conducted on melanoma. So of course, I've shown you data on lymphoma, but we have also data on melanoma and on breast cancer demonstrating the vaccine is highly versatile and could be applied to any kind of tissue, any kind of cancer, as long as you have access to the antigen derived from that cancer. So now I'm going to switch gears and talk about another type of vaccine where we can apply the Accum. So I've shown you the dendritic cell vaccine, which is a cellular vaccine. Now I'm going to talk about a protein-based vaccine targeting cervical cancer. So again, HPV is the virus that causes cervical cancer, and usually everybody practically is aware of Gardasil 9, which is a vaccine targeting the L1 proteins from 9 different subtypes of the HPV. So more specifically, HPV 6, 11, 16, 18, 31, 33, 45, 52, and 58. So if you develop an immune response against these HPV subtypes by triggering antibodies, and the HPV won't be able to transform a epithelial cell into an immortalized cancer cell and develop cervical cancer. So that's the idea behind the use of Gardasil 9. However, this vaccine could only be used prophylactically, meaning before the infection. Patients have to get the vaccine, or subjects have to take the vaccine before they get exposed to the virus. So if the patient develops cervical cancer or is being infected by another subtype that is not covered by the Gardasil 9 vaccine, well, in this case, that patient is going to develop cervical cancer, and there's no way you can treat it after that except using standard of care if that's feasible. So who are the competitors in the field? Of course, we know that Gardasil and Cervarix are being developed or manufactured by Merck and GSK, which are the giants in the field. There's only one company so far developing a therapeutic vaccine against cervical cancer, and that's Inovio. Their DNA vaccine is in phase III. And that's really where we're coming into play with our AccuVAC-POO7 based on the E7 oncoprotein. And by the way, this vaccine, as you can see, is not only, as you will see in a short moment, is not only targeting cervical cancer from a therapeutic perspective, but could also be used to protect from the establishment of the cancer. So it could be used both prophylactically and therapeutically. So we can basically touch two birds with one stone. So just to remind you, the genome of the HPV virus is composed, it's not a gigantic genome, so it has capsid proteins involved in growth stimulation, replication, and transcription. And of course, the two main oncogenes that are known are E6 and E7. So why these two oncogenes are quite important? Because these are the proteins that will immortalize a healthy epithelial cell to become a cancer cervical cell. So E6, for example, will block P53, which is an which is an apoptosis inducing protein, which usually leads to cell death if the cell is being infected or the cell feels that something wrong is happening. So the E6 protein will always block p53 activity, and the E7 can not only block p53, but can block other proteins involved in regulating growth arrest, such as p21, cyclin D, and RBE2F. So you can imagine that if a cell contains these two oncoproteins or oncogenes, then it will be transformed. It will lose its ability to die and will become immortalized, forming eventually cervical cancer. So what we did is we took our E7 protein, we added the Accum on it, and then we delivered it prophylactically. So before challenging with the cancer cell line, the C343. So the vaccine was delivered subcutaneously at day 0 and day 14. 2 weeks later, we challenged the animals with the cancer cell line. And what we noticed is that the animals that actually got the cervical cancer following the AccuVAC-POO7 vaccine never allowed the tumor to appear in these animals. And that despite 2 additional challenges that were done later on. So we did an initial challenge at day 28, which actually is equivalent to day 0 here. But then when we noticed that the tumor never grew, we challenged again the animals with 2 more injections of the C343, and these animals never developed tumors, again demonstrating that we have successfully achieved a powerful memory response. And if you look at the survival over time, when these animals all survived over 90 days, which is a very long lifespan, lifetime for these animals. So with these encouraging results, we then moved on to a therapeutic setting where we first injected the tumor, and then we delivered the vaccine at 2 shots, 1-week interval, along with different antibodies targeting immune checkpoints, or just to stimulate the immune response, or even to trigger what we call efferocytosis on cancer cells, which means that you have some phagocytes that will start attacking the tumor cells with the antibody use. And what we observed in this case is that if you combine our vaccine, for example, with the anti-CD47, which triggers efferocytosis against cancer cells, you end up having about 40% survival over 40 days. Now, that experiment was done, of course, where we used PD1, CTLA4, 4-1BB, and CD47. Of course, CD47 was the one that triggered the best effect, but right now we completed another study, where we used more than 1 microgram of the vaccine. We tested 5 micrograms, 10 micrograms, and with the use of the CD47, we were able to lower that tumor volume up to here, to very low level, with 100% survival. So right now we're conducting conjunction with PD1, CTLA4, 4-1BB, and CD47 to be able to demonstrate that the synergistic effect could be achieved with each one of these and immune checkpoint blockers or agonist antibodies. So that's work in progress, and we should be getting the results by the end of July. So again, this study demonstrates that there is synergistic effect between the vaccine and the CD47, for example, because if you look at CD47 here in the dotted line, we don't really see, we see an amplified immune response when the 2 are combined together, which again reflects the synergistic idea we're trying to establish with our product. So that product, right now we're finalizing the GLP study, and there is also a phase I trial that is planned early on in Canada next year, where we'll be testing the vaccine in a 3+3 design. We still haven't decided where exactly is the location of the phase I trial going to take place, but that's being actively looked at the moment as we speak. So with these different 2 examples of vaccination, so using a cell vaccine and a protein vaccine, I'm then going to switch gears and talk to you about another type of vaccine, or not vaccine, but another type of anti-cancer treatment, which is using the Accum molecule itself, a program we call AccuTOX-001. And that is a program that could be used against solid tumors with multiple indications before us. So how is the AccuTOX, how was it discovered? So initially, if you recall from the DC vaccine project, what we used to do is we used to take the Accum linked antigen, we would use that on resting DCs. So we pulse resting DCs with this formulation, and then we ended up having our DC vaccine. So then we asked ourselves, what would happen if you take, for example, the antigen and you mix it with the Accum, and then you add that mix on resting DCs? So instead of chemically linking these two molecules, you would just mix them together. And so what we noticed then is that all of the DCs died. So the cells couldn't actually survive after 3 to 6 hours treatment with this mix. So that made us wonder, is the Accum actually toxic when added directly on the cells? So using the EL4 T-cell lymphoma, we conducted an IC50 dosing. We were looking at percentage of survival of these cells treated with the Accum molecule, and we noticed that indeed the Accum molecule was able to kill these EL4 tumor cells, but of course the IC50 was quite high, was about 95.94 micromolar. Usually a drug becomes appealing when it's in the low micromolar ranges or in the nanomolar ranges. So then with the help of our CTSO, we generated a large library of different variants of the Accum. So that's another advantage of this technology, in the sense that you can play with the nuclear localization sequence or the bile acid type, and you can generate a panoply of different molecules. And when we screened our library, we identified another molecule called an AccuTOX-001 with an IC50 of 3.2 micromolar. So in this case, the molecule was highly toxic compared to the original Accum. And here, I mean, the reason why the sigmoidal curves are just flipped is because here we're looking at the percentage of survival, and here we're percentage of cell death. It's exactly the same thing. You just have to flip them. But the take-home message is that the AccuTOX-001 is 30-folds better than the original Accum. So to convince you of that, we took the original Accum molecule, we tested it against T-cell lymphomas, colon cancer, melanoma, and breast cancer. And as you can see here, depending on the cancer type, the percentage of cell death could vary between 93% and 40% type thing. But if you take these same cell lines and you treat them with the AccuTOX, and here we use 2 different doses, you can see that you end up having 100% cell death, no matter what the cell type you're using is. By the way, we tested the AccuTOX on over 9 different cancer cell lines, both human and murine. We tested breast cancer, glioblastoma, non-small cell lung carcinoma, melanoma, and in all cases, the AccuTOX was able to actually kill those tumor cells. So we conducted a proof of concept in vivo. We transplanted tumors in animals. So this is an example done with lymphoma, but we have the same example conducted in melanoma and breast cancer. And as you can see, if you deliver the AccuTOX-001 alone as an intratumoral injection directly to animals, you can see here the tumor growth over time, with about 60% survival reaching day 40. Now, if you combine the AccuTOX with anti-PD-1, anti-CTLA-4, anti-CD47, which are immune checkpoint blockers, there you can see that you improve dramatically the anti-tumoral effect you can reach with this molecule. And in fact, we have data now to demonstrate that the AccuTOX is not only killing the cells, but it's also inducing what we call immunogenic cell death. In other words, when the tumor is dying, it releases signals that activates the immune system to mount an immune response against the tumor itself. So there is an immune component involved even with the cell death induced by AccuTOX. And as you can see, when for example, you deliver AccuTOX with anti-CD47, you end up having 100% survival, exactly the same way as if you're combining with anti-CTLA-4. So, when we looked at this molecule with the melanoma, it was mostly the PD-1 that led to 100% survival. When we looked at cervical cancer, breast cancer, it was mostly the CD47 and CTLA-4 that had a good effect. So depending on the tumor type, the immune checkpoint blocker could change, which is normal. But the take-home message is that the AccuTOX alone or in combination with immune checkpoints could lead to a potent anti-tumoral effect and could eventually treat subjects with cancer. So again, through a synergistic effect. So this is the 3rd phase I trial we would like to eventually initiate. So hopefully in Q1, Q2 of 2023. It's a dose escalation study, again, a 3+3 design in combination with immune checkpoint blockers. Could be PD-1, but we can also test other immune checkpoint blockers depending on the final indication we will be choosing after the ongoing negotiations. So now that I've spoken about two different vaccines, one cellular, one protein. I've spoken about the AccuTOX, which is a small molecule Accum variant that could be used as a direct treatment for cancer. I'm going to show you now how we can also use the Accum against in the context of an antibody-drug conjugate, which is a very big program of defense. So the antibody-drug conjugates, or ADCs for short, are composed of three main components. So you have the antibody itself, which targets a specific molecule on the cell surface of a tumor cell. You have the linker, which basically will attach a drug called the payload on the antibody. So you have the antibody, the linker, and the drug. So this is forming the antibody-drug conjugate. Now, the problem with the ADCs is that most of the time they can reach their target site. That's fine. They get endocytosed like any other biomolecule, but then they get entrapped within the endosomes, and therefore that will limit the drug diffusion outside of the endosome, where then you can start developing mechanisms of resistance or inefficacy towards your intracellular target. So what we said to ourselves, we said, well, in this case, the Accum could be linked to an ADC, and there it can break down the endosome, allowing the drug to diffuse in a much efficient way and eventually reach its subcellular target. So we applied that to trastuzumab DM1, called the ADC-001, and we also applied it to another antibody-drug conjugate, the trastuzumab linked to alpha-amanitin. And when we tested, for example, the trastuzumab DM1 or Kadcyla on two different breast cancer cell lines, we noticed that the ADCs that had the Accum molecule, they had 10 to 100-fold better efficacy against these tumor cell lines. And that's shown by these sigmoidal curves are shifting towards the left. So that means you need less of the antibody to be able to reach a cellular cytotoxicity or apoptosis of cancer cells. Similar effects were observed with the cleavable trastuzumab linked to alpha-amanitin, where you see clearly here the Accum linked T-alpha-A, leading to a much better therapeutic effect. So these were some of the preliminary work we done with the Accum, but of course we moved forward and we tested a variety of different Accum variants with these antibodies. This is done through a collaboration with the HUS Cancer Center in Helsinki. And that step was actually completed, and we have here examples of screening conducted with different variants where we detected the most efficient ones. And right now, the lead compounds, the lead ADCs were selected and they were sent to the Curie Institute in France to be tested in what we call patient-derived xenograft models. So these are animal models that were basically these, they're deficient mice that are grafted with patient-derived tumor cells that were then treated with the different lead ADCs that we have. So we're expecting the final results by the end of the summer, so most likely in August, so we can eventually have our lead compound and move forward down the line. So of course, I don't have to remind you that this is a very big field. It's a multi-billion-dollar field combining the big players, big pharmaceutical companies. This is just some of the competitors in the field, but technically every single pharmaceutical company has an ADC somewhere in development. So the implications are humongous in that field for defense, of course. Now, I hope I was able to actually convince you through this general overview of the versatility of this technology. So I've shown you that you can easily apply it to the design of a DC vaccine. So we conducted an example with the T-cell lymphoma. But just remember, if you have any antigen in your hand, any tissue sample coming from a specific cancer, that vaccine could be applied to. So this is not a melanoma-specific vaccine or a lymphoma-specific vaccine. It's a vaccine platform that could be applied to any cancer type as long as you have access to tissue samples. You could actually link the Accum to any protein of interest. We've shown you, for example, the E7 oncoprotein, but we've done it on spike. You could do it on any protein derived from malaria, leishmania. You can do it for let's say monkeypox. Any protein that comes to your mind becomes easily linked to Accum to be able to be formulated and tested as a vaccine. We've shown you that Accum could be modified to generate a variant with anti-cancer properties. I've also demonstrated some examples of currently available FDA-approved ADCs that could be improved tremendously with the use of Accum. And of course, the company is also working on developing its own in-house antibody-drug conjugate with its own payload. Because if you think about it, if you develop an antibody, an ADC, well, you also know that Accum could become an anti-cancer molecule. So then you can link this anti-cancer molecule to the ADC that you have, and you can improve not only the endosome breakage to induce the escape, but also improve the toxicity mediated by the payload. So, and of course, what you have to keep in mind is that in all these different conditions, including the ADCs, you can achieve synergistic effects with the immune checkpoint blockers that are currently being used as standard of care in cancer patients. So if you have any questions in the future, please feel free to reach us by email or even call the CEO. I encourage you also to visit our website, defensetherapeutics.com. You can learn more about us, but I encourage you to go to the Accum technology platform, where there we really cover up the rationale behind this technology. Where does it come from? What's the objective of using it? So really accumulating the biomolecule directly in target cells, whether it's an ADC, RNA molecule, DNA molecule. We even have data on CRISPR. So it really shows you what are the next steps we're following on, where we are exactly in terms of our development. So this website is actually updated continuously. And you will probably see also our press releases, vaccine development objectives, where we are right now with the COVID, for example, the cancer vaccines. We talk about, our future plans in the sense of DNA and RNA vaccines again, and talk to you about the timeline we use to achieve our objectives. So with this, thank you very much. And please feel free to get in touch with us again by email or by phone call if you have further questions.